Welcome to Spring LIB.
Hello everyone, today I'll be helping you with the 70th problem of the chapter 14 problem set. So you have A that is asking you to, as described in 14.3, transformation, or HGT, was reported by Griffith in 1928 in an experiment in which of the following occurred. So what you want to know is that you have the heat pathogen with the stuff in it was heat treated. And then you have a non-pathogenic thing. Basically what happened is that the stuff went into the pathogen and killed the mouse. So if you look at all of them, one says heat-treated pathogenic bacteria recovered their pathogenicity when incubated with non-pathogenic bacteria. Then you have two, plasmids were transferred to non-pathogenic bacteria, which is false, so two's not it. You have three, non-pathogenic bacteria acquired pathogenicity when incubated in a broth containing heat-treated pathogenic bacteria. Then you have four, so no, it's not two or four. And then what happened is that it is three, because one says that the pathogenic bacteria recovered their pathogenicity, which is false. So then you have B, which is asking you Griffith's. So basically what happened in the Griffith's experiment, and basically it's asking you what was observed in the Chase and Hershey experiment. So then you know that the bacteriophage culture, as in 4, grown in a nutrient-containing medium and radioactive, labeled using sulfur isotope, did not transfer to the bacteria incubated in an unlabeled nutrient containing. So that is true. So it is 4. And then you have C, which is transformation and transduction, increased variation within populations of bacteria, and RK bacteria by which of the following. So you have to basically figure it out. So you have A, transferring DNA among different species, which is true. That is actually it. And then you have B that says transferring free DNA across the cell membrane, which is false. You have C that says transferring DNA between different strains of the same species, false. Then you have D, phagocytosis of bacteriophages. So then you have D that says identify a factor that might affect transformation, or HGT, then design a plan to evaluate the dependence of how it'll happen. So for me, I would do pH, and then I would see if the denaturing of the protein made it so that the information cannot transfer to a non-pathogenic. So it's very similar to this experiment, except this one would be treated with a very low pH, and then it would be mixed with a non-pathogenic one and see if the DNA or that the pathogens transferred to the non-pathogenic one. So I hope you found this helpful, and I hope you have a great